Hi everyone, how are you? Have you ever wondered how scientists can differentiate the variants of COVID 19 and how it spreads across the globe? How are scientists able to develop several vaccines for COVID 19 and how do they differ from each other? Not only for COVID 19, how are drugs being developed to target certain types of diseases? How are scientists able to validate the efficacy of these drugs? On crops, on the other hand, have you ever wondered why some types of crops are more resilient to drought than other crops? Or why do they produce a better quality despite the changes in their environment? Which gene is responsible for developing resistance to pests that destroy their yield? Now, look at your family or friends. Have you ever wondered why most of us in the Philippines have brown eyes and naturally black hair? Why don't humans look the same? Despite that, each human DNA is 99.9% similar. Will the 0.1% dissimilarity help us understand human evolution? What genetic markers help scientists to identify who among us are more likely to be susceptible to certain types of diseases? And how do we respond to certain types of medicines? These are just some of the questions bioinformatics is trying to make sense of. As you may see, all of these applications are related to biological data, may it be genes, genomes, RNA, proteins, and metabolic processes, all essential information that make up the main components of life. So, okay, you get the bio in bioinformatics, but what about informatics? As you may have thought, with this computer age and the advancements of technology, biological information has been made readily available for us, and the studies mentioned earlier generate voluminous amounts of data. Here's another analogy for you. I know all of you are familiar with the jigsaw puzzle. You may have played with as little as 10 pieces. They are easy to put together, right? But have you ever tried assembling 10,000 jigsaw puzzle pieces? This may take a while for you to put them all together alone, or you may need to tap friends and family members to help you piece the puzzle together to beat your time record of solving when you only had 10 pieces. If there were more pieces, maybe you'd wish to work with computers and with artificial intelligence. This idea is why bioinformatics came to be. Due to the large amount of biological data that researchers have to deal with, around 3 billion base pairs for the human genome alone, by the way, experts from several different fields like computer science, applied mathematics, statistics, biology, chemistry, all come together to make sense and deal with the data. That's why bioinformatics was born. Bioinformatics is a multidisciplinary field that uses computers and statistics in developing software tools and algorithms to analyze large data sets of biological data such as DNA, RNA, and proteins. To understand more about data availability, let us visit a short history of sequencers. Although DNA was first discovered and isolated by Friedrich Mischa in 1869, it was not until the 1950s when Frederick Sanger laid out the foundation for sequencing proteins that paved the way further for DNA sequencing. In fact, it was only in the late 1970s when the first full DNA genome of bacteriophage Phi X174 was sequenced. This event marked the beginning of what we now call first-generation sequencing. The most common of all first-generation sequencing utilizes the chain termination sequencing method developed by Sanger. These first-generation technologies were initially done manually and were later automated. The first-generation DNA sequencing machines produce reads slightly less than 1 kilobase in length. That's roughly about 6 to 7 single text messages. It also has low coverage of $500 per megabase, or roughly about $500 per 6,000 to 7,000 single text messages. The runtime takes months to generate, and it has low accuracy. However, these disadvantages of the Sanger method inspired advancements to several new technologies. To analyze longer fragments, Researchers made use of shotgun sequencing, where overlapping DNA fragments were cloned and sequenced separately and then assembled into one contiguous sequence. This marked the beginning of second generation sequencing and paved way to the next generation sequencing. The technology behind next generation sequencing, in contrast to first generation sequencing, is characterized by being highly scalable that allows the entire genome to be sequenced at once. In 1990, the Human Genome Project officially began. At this point, first generation sequencing is still widely used. However, by 2001, shotgun sequencing technologies were incorporated and a draft sequence of the human genome was produced, which led to the project's completion in 2003, way ahead of schedule. These next generation sequencers allowed reading of DNA hundreds of base pairs in lengths that are massively parallelized to produce gigabases of data in one run. That's roughly around 6 to 7 million single text messages per run at a cost dramatically lower than its predecessors. To give you an example, according to Illumina, the HiSeq X10 will yield whole human genome sequences for $1,000 each whole genome and will have the capability to generate around 15,000 to 2,000 genomes per year. To this day, third generation sequencing technologies are constantly being developed. Third generation technologies aim to increase the rate at which samples can be processed, decrease the time to obtain a result, and at the same time decrease the cost further by eliminating the need for excessive reagents. Some of these devices are even as small as your mobile phone. The MinION by Oxford Nanopore Technologies is capable of producing 50 gigabases of data per MinION flow cell, or roughly 3 million text messages for only $1,000. The advent of rapid DNA sequencing methods, with a dramatic decrease in the cost per sequencing, has greatly accelerated biological research. One cannot further emphasize the need for bioinformatics.
Now that we are acquainted with what bioinformatics is and what questions it can answer, let us look deeper into the core concepts when starting out with bioinformatics, and these are the file types. One of the nuances of being in a bioinformatics study is trying to make sense of the varied file types. But before that, let us get to know what file format is. A file format is a way for computers and humans to standardize how data is organized. Additionally, computers can check file formats and immediately determine whether it should be opened in a text editor, a browser, or some other specialized software. Take for example this file. This file can be opened and easily readable in a Word editor, specifically a Microsoft Word program. If you try to open the same file in a spreadsheet reader or MS Excel, the program might not be able to open nor present the contents of the file in the best format that it was originally intended to. If you open this image file on the left using a video editor program, it might not be able to open it. What we are saying is that file types are usually software or program specific. However, some are cross-platform. In terms of bioinformatics, examples of these are FASTA, AB1, and FASTQ file types, among others. Let us now see the various file types used in bioinformatics. We will look at their uses and how they differ from each other in terms of content and format. The FASTA format was invented in 1988 and designed to represent nucleotide or peptide sequences. It originates from the FASTA software package, but is now a standard in the world of bioinformatics. A FASTA file contains sequence data along with the header. A FASTA file starts with the greater than symbol. It is followed by a header, which is a string of characters that can be used to describe the sequence. It could contain the sequence ID, description, function, locality, sampling site, among others. The sequence, on the other hand, is a sequence of nucleic acid or protein encoded in standard one-letter code. Notice that in this FASTA file, one is allowed to concatenate several sequences in one file. The FASTQ format was developed by Sanger Institute in order to group together sequence and its quality scores, or the Phred quality scores. In FASTQ files, each entry is associated with 4 lines. Line 1 begins with the @ character. It also contains the sequence identifier and an optional description. Line 2 is the sequence in standard 1-letter code. Line 3 begins with a plus character and is optionally followed by the same sequence identifier or any additional description again. Line 4 encodes the quality values for the sequence in line 2, and it must contain the same number of symbols as letters in the sequence. Similar to the FASTA file, a FASTQ file can have several sequences in one file. Let us get to know what a Phred score is in a FASTQ file. It is a quality score or Phred scale for each base pair. It indicates how confident we can be that the base was sequenced and identified correctly. Phred scores use a logarithmic scale and are represented by ASCII characters, mapping to a quality usually going from 0 to 40. The quality, also called the Phred score, is the probability that the corresponding base call is incorrect. P here is the probability that the corresponding base call is incorrect. For example, if the Phred quality score is 40, then the probability that the base is called wrong is 1 in 10,000. Another way of interpreting it is that we are 99.99% sure that the base was called correctly. Now, if the Phred score is a number from 0 to 40, why don't we see numbers in line 4 of the FASTQ file shown earlier? That is because rather than giving numeric values of Phred score, they are provided in ASCII character codes from 33 to 126. Now why 33 to 126? Because 33 to 126 codes for single characters. FASTQ Sanger utilizes score from 0 to 93, while FASTQ Illumina uses scores from 0 to 62. The 0 in each Phred score maps to ASCII character 33 or the exclamation point. If the Phred score is 90, then it maps to ASCII character 123 or the left curly brace. Why don't we try an example? Can you tell me the accuracy of calling the base C in this sequence? The ASCII character equals maps to the Phred score 28. If we go back to our formula and plug in 28 to Q, we will get a value of 0.001584893 for P. How do we interpret this? Subtracting it from 1 means that we are 99.84% sure that the base C was called correctly. Now let us see who among you is listening to this lecture. Answer this. What is the accuracy of calling the base G in this sequence? You might want to get a pen and paper to write some information. Here are the ASCII characters. And here is the formula. You may pause this video and compute for the accuracy of the base call. What do you think is the answer? How about this one? What is the accuracy of calling the base C in this sequence? Here again are the ASCII characters. And here is the formula. Go ahead, try it for yourself. The SAM file is a text-based format for storing biological sequences aligned to a reference sequence. It was developed by Heng Li and Bob Handsaker. It was developed when the 1000 Genomes Project wanted to move away from the MAQ mapper format and decided to design a new format. SAM stands for Sequence Alignment Map. The header line starts with the @ symbol, while the alignment lines do not. The header holds generic information on SAM file along with version information, if the file is sorted, information and reference sequence, among others. The alignment records constitute the body of the file. Each alignment line or record has 11 mandatory fields describing essential alignment information. If the information represented by any of these fields is unavailable, that field's value will be a placeholder, either a 0 or asterisk as determined by the field's type. In this example, this one here refers to the query name. This refers to the reference sequence name of the alignment. This here is the sequence segment, while this one here refers to the ASCII of base quality. A BAM file or binary alignment map is a compressed binary version of the SAM file. It is a compact and indexable representation of nucleotide sequence alignment. The data between SAM and BAM files are exactly the same. Being binary, BAM files are small in size and ideal to store alignment files. VCF stands for Variant Call Format. It is used to store gene sequence variations. The format has been developed for genotyping projects and is a standard to represent variations in the genome of a species. The header contains meta-information and is included after the double hashtag string. It is recommended to include info, filter, and format entries for a better explanation of the data field. The data lines have 8 mandatory columns. It includes an identifier from the reference genome, the reference position with the first base having position 1, an identifier, a reference base or bases, among others. The general feature format is another text file format used for describing genes and other features of DNA, RNA, and protein sequences. It is the standard for annotation of genomes. A GFF file should contain 9 columns. The columns include the name of the sequence where the feature is located, the keyword identifying the source of the feature, the feature type name, the genomic start and end of the feature, and so on. Let us move to a more global picture of bioinformatics involving databases. To get an information, we have to get that file with a particular file type on the network that we have. The first layer of the network is our own workstation. This workstation is the first level of storage of this information. A collection of these workstations connected together is what we can consider a server. If we can connect the various servers using the internet, then users from various geographical locations can now have access to information from various geographical locations. Each of these servers may house a specific type of file. Example would be a server which only files DNA sequences, and then another server may specialize in protein sequences, and so on. Databases on bioinformatics include databases housing RNA sequence data, DNA sequence data, and protein sequence data, among others. The National Center for Biotechnology Information is the most extensively used and accessed database. It is housed in Bethesda, Maryland. It was founded in 1988 through legislation sponsored by Senator Claude Pepper. It is a repository for nucleotide, protein, domains, diseases, whole genome assemblies, transcriptome assemblies, and more. This database does not only contain information generated in the United States. Any researcher may contribute to this database. Here is an example of searching from the NCBI database. The matching query will provide you information on literature, genes, proteins, and genomes, among others. The Kyoto Encyclopedia of Genes and Genomes is a collection of databases dealing with genomes, biological pathways, diseases, drugs, and chemical substances. The KEGG database project was initiated in 1995 by Minoru Kanehisa, professor at the Institute for Chemical Research, Kyoto University, under the then ongoing Japanese Human Genome Program. Here is an example of how to search in the KEGG database. Here you can see the pathway map of a fatty acid biosynthesis. The authors and journals related to this query are also shown here. The pathway map shows the enzymes or the products of the fatty acid biosynthesis. UniProt is a freely accessible database of protein sequence and functional information, many entries being derived from genome sequencing projects. The UniProt consortium comprises of the European Bioinformatics Institute, the Swiss Institute of Bioinformatics, and the Protein Information Resource. The UniProt consortium was organized in 2002. This is an example of how we can search using UniProt. Matching results from the query are listed in rows. When you click on each result entry, you will be given further information on the function of the protein. Taxonomy is also provided. The interaction with other proteins are also available. A 3D model of its structure is conveniently placed on this page as well. You can interactively zoom in and out and rotate the structure. A plethora of other information is given as well. ExPSI stands for Expert Protein Analysis System. It was one of the first websites of the life sciences and among the first 150 websites in the world. It is an extensible and integrative portal which provides access to over 160 databases and software tools. Essentially, ExPSI database is the database of databases in bioinformatics. ExPSI supports a range of life science and clinical research areas, from genomics, proteomics and structural biology, to evolution and phylogeny, systems biology, and medical chemistry. These are examples of how to search from inside ExPSI. When you search for UniProt, it will provide you with a link to the UniProt database and other related databases. You can also type BLAST and it will show you the results related to local alignment tools. NCBI has an implementation of the tool BLAST. BLAST stands for Basic Local Alignment Search Tool. It is an algorithm for comparing primary biological sequence information, such as the amino acid sequences of proteins or the nucleotides of DNA and/or RNA sequences. A BLAST search enables a researcher to compare a subject protein or nucleotide sequence with a library or database of sequences. It also identifies database sequences that resemble the query sequence above a certain threshold. Let us try to use BLAST on a nucleotide sequence. I have in here a partial sequence from the complete genome of SARS coronavirus from Urbani. Let us paste it into the NCBI BLAST text field and see if it matches the SARS-CoV-2 sequence of Urbani in their database. The time it takes to search their database is proportional to the length of the input sequence. Since the sequence we provided was not that long, it shouldn't take long. Notice that the SARS-CoV-2 from Urbani had the most significant alignment with our query sequence. You can inspect each match further by clicking on each row. That sums it up for databases. There are more databases available, however, the ones shown here are the most widely used.
Now, let's move on to bioinformatics pipelines. Pipeline transport is the long-distance transportation of a liquid or gas through a system of pipes called a pipeline. It is typically sent to a market area for consumption. The idea of a bioinformatics pipeline, or alternatively called workflow, is similar to the literal meaning of pipelines. However, in this case, what is being transported is data. It is evaluated at each section of the pipeline geared towards answering a biological question for the consumption of concerned groups or community. The data that came in at the start of the very first pipe in the line may not be the exact data at the very end of the line. The data has already been analyzed and transformed into a more usable and insightful version of its initial information. Perhaps one of the most important concepts in data science, including bioinformatics, is the GIGO principle, or the garbage in, garbage out phenomenon. Garbage in, garbage out, or GIGO, is a truism for any informational system reflecting the idea that poor quality input will produce a faulty output. If you don't clean the data, or if you hide about the state of the data, you will probably generate results that are not applicable or reproducible in another similar situation. Related to the GIGO principle is the large amounts of data generated by next-generation sequencing technologies. This has created important challenges for biological research. As a matter of fact, during experiments, issues often emerge in library preparation or in the sequencing of the data. Hence, the very first line of defense against producing garbage in bioinformatics is the quality control step. The bioinformatics team should take into account the experiment environment, sample time points, and scenarios. By doing checks, poor quality data can be isolated. Conducting sequencing quality control protocols at different moments of data processing, as well as correctly interpreting the quality control results, are crucial steps to ensure a successful and meaningful study. Some NGS quality controls to increase the accuracy of the outcome are the following: raw sequencing data quality and depth, alignment quality, and reads duplication rates, among others. The analysis part of the pipeline can involve several other steps or branches. Here are a few examples. From a single sequence, one can find similar sequences from various databases. From the list of these similar sequences, this can be further aligned to many other sequences or directly evaluated for sequence feature and function. From the multiple alignment result, a phylogenetic tree can be visualized, or the annotated sequence can be visualized as well. Here is an example of a workflow to identify potentially pathogenic variants taken from an exome. After mapping the sample exome to the reference human genome sequence, variants are determined to know if there is a difference versus some reference at a given position in an individual genome or transcriptome. These variants are then annotated to assign functional information and identification of single nucleotide polymorphisms or genetic variation. The mutations with significant variation can be viewed as a 3D structure biological molecule. Let us look at the very simple workflow in bioinformatics. The alignment of biological sequences is probably the most important and most accomplished in the field of bioinformatics. Alignments are a powerful way to compare related DNA or protein sequences. They can be used to capture various facts about the sequences aligned, such as common evolutionary descent or common structural function. We take the general view that the alignment of letters from two or multiple sequences represents the hypothesis that they are descended from a common ancestral sequence. A superposition of the template structures is shown here. After a successful alignment has been found, an actual model building can start. Since every person inherited DNA from their parents, who inherited it from their parents, and so on, a person's DNA is made up of the DNA of their ancestors. If you and another person both have the same ancestor, there's a chance that you both inherited some of the same DNA. All of these examples can be accomplished by the workflow on the left. Once a pipeline is established, you may either run your pipeline through a graphical user interface or via a terminal. Graphical User Interface, also known as GUI or GUI, allows users to interact with the computer through graphical icons or audio indicators, while terminals are purely text-based applications that take in commands from the user and directly pass them to the computer's operating system. Other terms for the terminal are shell or command line. GUIs are user-friendly and most advantageous for the visualization of your data. However, GUIs take up a lot of memory space. This is why most bioinformaticians create their codes or scripts that implement their pipeline on a terminal. Apart from eliminating the need for additional mouse clicks that increase the time to run the data, using the terminal allows the bioinformatician to allocate additional memory space for running the data instead of the aesthetics you get from a GUI. On the downside, the terminal requires one to learn scripting and the basics of the command line using the Linux operating system. In this training, you're in luck because we will be teaching you the basics of navigating the Linux command line to let you feel how it is to become a bioinformatician. So why do we really need pipelines or workflows? First and foremost, pipelines generate a standard process for a bioinformatician's task. A workflow divides the entire process into modules. When one part of the workflow fails, we can check the faulty step and recover the files or restart the analysis at that checkpoint without having to run the entire process again. Pipelines also allow parallelization of data analysis. A single set of data output can be analyzed through a series of simultaneous tasks independently executed from each other. At each step of the pipeline, we can know which software to use, and at each type of file, you can also pin versions of the software to use for that particular file that is compatible with the data. Now that you are acquainted with the basics of bioinformatics, you are now ready to begin with the first part of this science immersion program, learning the basics of the Linux command line. We hope that you will enjoy this workshop made especially just for you.